In the complex, shifting waters of the lower Danube and the Black Sea coast, toxic algal blooms are expanding rapidly. These biological outbreaks choke aquatic life and degrade water quality, requiring immediate, scientific mitigation. Standard visual inspection from the shore, or even basic RGB aerial photography, cannot provide the data needed to intervene. To human eyes or a standard camera, we only see a surface discoloration. Effective environmental defense requires exact, quantitative estimates of the actual algal biomass beneath the surface, rather than a simple confirmation that green water exists. To solve this, the research team at UDJG engineered a comprehensive data pipeline, a continuous workflow that translates physical ecosystem markers into predictive digital models. The first stage of this pipeline relies on specialized hardware. A UAV is deployed to capture specific photons reflecting off the water using a 5-band multispectral sensor. The second stage grounds this aerial data in physical reality. The flight captures are synchronized with precise in-situ philometry measurements taken directly from the water. These synchronized arrays of aerial and physical data are then fed into a custom convolutional neural network designed to automate the detection of bloom outbreaks. Solving environmental monitoring at scale is a systems engineering challenge. It requires a tightly coupled pipeline where the rigid physics of optical sensors directly dictate the success of the digital machine learning architecture. The accuracy of the machine learning model is strictly limited by the physical parameters of the data capture. The most critical metric here is ground sample distance, or GSD. GSD defines the physical area of a single pixel. As a drone decreases altitude, the spatial footprint on the water shrinks, capturing a denser area. This is governed by calculating flight altitude, camera focal length, and sensor resolution. Achieving a sub-meter ground sample distance is a technical requirement for this pipeline. The granular, microscopic structures of early-stage algal bloom initiation are entirely lost if the pixel represents too broad an area. This creates a strict physical constraint. Lower flight altitudes yield the high-resolution data the algorithm needs, but they drastically reduce the territorial coverage mapped per flight. To maximize the data gathered within those flight limits, the drone is equipped with a specific sensor payload, the RedEdge MX camera. Standard cameras compress light into a basic red, green, and blue image. The RedEdge MX bypasses this limitation by capturing data across 5 distinct narrow spectral bands simultaneously. 2 of these bands, the near-infrared and the red edge, are highly sensitive to the optical signatures of chlorophyll, allowing the sensor to detect biological activity invisible to the human eye. Without mathematically optimizing the ground sample distance and capturing targeted 5-band spectral data, the data fed into the algorithm is blind to the underlying biology. Down at the water level, researchers deploy a specialized probe known as the algae torch to establish physical ground truth. At this stage, the drone imagery consists solely of uncalibrated spectral reflectance values. The system needs physical, measured biomass densities to teach the algorithm what those specific reflectance patterns mean. During intensive monitoring campaigns in August and September 2024, teams systematically extracted these baseline florimetric measurements across the ecosystem. The next step is spatial synchronization. The exact spatial coordinate recorded by the algae torch is mapped directly onto the drone's orthomosaic. The pixel values representing the 5 spectral bands at that precise coordinate are extracted and mathematically paired with the in-situ biomass density. This process also demands tight temporal synchronization. Because river currents constantly shift the water's surface, the drone flight and physical sampling must occur simultaneously to prevent data drift. The output of this phase is a highly structured data matrix, a direct row-by-row pairing of the 5-band spectral signatures with verified physical biomass quantities. This precise spatial and temporal synchronization acts as the primary bridge linking the physical mechanics of the ecosystem to the digital architecture of the predictive model. Initial algorithmic tests applied standard global machine learning regression models to this synchronized dataset. These global models struggled to perform accurately. They failed to account for the complex spectral variations introduced by differing flight altitudes and shifting sunlight conditions. To process this highly variable data, the architecture required a pivot to deep learning, specifically deploying convolutional neural networks, or CNNs. A CNN feeds a 5-band image patch into a convolution layer, where a 3x3 filter systematically slides across the grid. This extracts complex feature maps, isolating spectral gradients. The data then passes through a pooling layer, reducing spatial dimensionality without losing vital structural features. This is a chart comparing the classification accuracy of the models. The results demonstrate a distinct leap in performance when using the full 5-band multispectral data over a standard 3-band RGB baseline. To further refine the model, engineers implemented specific spectral normalization strategies, standardizing the data to correct for the inconsistencies caused by data captured at varying drone altitudes. The final system employs a dual model architecture. One CNN operates solely to classify whether bloom initiation is present, while a secondary model processes the positive hits to estimate the exact biomass density. These convolutional neural networks actively learn the spatial and spectral geometry of biological organisms, allowing them to outperform traditional algorithms that passively read pixel colors. A predictive model has no practical value if locked inside a data scientist's environment. The tool must be operationalized for deployment in the field. This leads to the final deliverable of the project pipeline, a decision support system packaged as the HAB 5-band final app. The user flow is highly automated. The application begins by ingesting the raw 5-band UAV orthomosaics directly from the flight payload. Once uploaded, the software executes the pre-processing invisibly in the background, calculating the necessary spectral indices before feeding those data arrays into the trained CNN. The application outputs a clear, actionable result, a quantified spatial map of the ecosystem pinpointing exact algal density. Environmental monitoring agencies rely on this specific spatial output to execute immediate, data-driven decisions and deploy targeted mitigation strategies. By mathematically linking drone flight physics, rigorous in-situ fluorometry, and 5-band neural networks, this pipeline successfully processes raw environmental photons into an automated, highly effective defense mechanism.